Alright, welcome to part 2 of Biomolecules, which involves proteins, enzymes, which are specialized proteins, and nucleic acids. You'll be using fill-in-the-blank notes for this part 2 lecture. And at the end of the lecture, I want you to go ahead and draw a Venn diagram comparing carbohydrates, lipids, and proteins. We'll leave the nucleic acids off of this one for now. You'll be doing another graphic organizer later next week to review over all 4. Turn that Venn diagram into the box before you leave, and we'll put it in your journal when I give it back to you on Friday. Okay, so proteins. What are proteins? Proteins are large molecules that contain carbon, hydrogen, oxygen, and nitrogen, or as we like to say, CHON. Okay, they are found in every cell in the body. They are involved in most of the body's functions and life processes. The sequence of amino acids, which is their monomer, is determined by DNA. So the DNA determines which sequence of amino acids goes into the RNA to help build and make a protein. We'll learn about that later. So again, the monomers, mono meaning one, mer meaning part, is an amino acid. Amino acids are compounds with an amino group on one end and a carboxyl group on the other end. The polymer, meaning many parts, poly meaning many, mer meaning part, is a polypeptide chain, which is, again, just a fancy word for proteins. So one single amino acid is the monomer. A chain of amino acids, or a polypeptide chain, is a protein. It's found in every cell in the body and in foods such as beef, chicken, fish, beans, and nuts. There are more than 20 different amino acids found in nature. Amino acids are joined by covalent bonds. The instructions for arranging amino acids into many different proteins are stored in DNA, or our deoxyribonucleic acid, which is another type of biomolecule. All right, so the functions of proteins: control the rate of reactions, meaning enzymes. They act as catalysts, and we'll get into that in specifics here just a little bit. They regulate cell processes, so hormones, so adrenaline, your fight or flight. Those are all regulated by hormones, okay? Transport substances into or out of cells. Transmembrane proteins, I spoke about that in the first part where we talked about the phospholipid bilayer of the cell membrane. It has proteins embedded in it called transmembrane proteins that help substances that are too big to get through that membrane go ahead and cross. It helps fight disease or antibodies. There's a picture of that over to the right. And it helps grow and repair tissue. And it provides structural and mechanical support, because it helps build muscle as well. An example would be hemoglobin. Hemoglobin is protein needed for oxygen to transport in blood. So if you don't have enough hemoglobin, then you're going to have a hard time transporting the oxygen to the rest of your body where it needs to go. All right, enzymes, those special proteins. Enzymes are known as biological catalysts, which means they speed up the rate of chemical reactions. So all biochemical reactions require enzymes so that life can exist efficiently. Reactions like breaking down and digesting food, building of essential cellular components, and the breaking down of bacteria and harmful toxins. All right, so Enzymes, again, are catalysts, and their cells use these to speed up chemical reactions. Enzymes speed up reactions by lowering what we call activation energy. The activation energy is the amount of energy needed to initiate or start a chemical reaction. Generally, they end in ACE or ASE. So anytime you see, just like with carbohydrates or sugars, they ended in ose, like glucose or lactose. Well, if you see one that is lactase, that means it's an enzyme that breaks down lactose. Specifically, you have what we call an enzyme for a substrate, enzyme-substrate complex. In order to catalyze reactions, the enzymes must bind to the reactants or substrate. So if you look at this picture right here, here is my enzyme, and here is my substrate. So this is the reactant that I'm wanting to break down at the active site. The active site has a very specific shape that is specific only to that substrate. So again, the reactants or the substrates bind and thus facilitate the formation of a product. The enzyme is not changing as a result, and it is immediately able to catalyze another reaction. So this enzyme comes in, it attaches to the substrate, or the substrate attaches to it at the active site. Bonds in substrate are weakened by the enzyme, and then the enzyme breaks up. Then the substrate breaks apart and becomes the products, so a new substance. The enzyme then is immediately able to go back and break down another substrate. All right, so enzymes work by lowering the activation energy needed to start a chemical reaction. What it does is a small amount of energy input that is required so the reaction can begin. So it lowers that activation energy needed. Factors that affect enzyme activity are temperature, pH, salinity, enzyme concentration, substrate concentration, and or the presence of inhibitors or activators. So if you look here at this graph, it says active energy profile of a reaction aided by an enzyme. So here are our reactants, just like the substrate. That's your substrate. I'm sorry. My goodness. That's our substrate right here. And the red is the reaction that happens without an enzyme. So this is the amount of energy required to start the reaction. So it goes, it's building up, and it finally gets that energy, right? So now it's reached its peak, it can activate and start that process of breaking down that substrate into the products that we needed. Okay? Then this is the energy released by the reaction right here. Okay, once it reaches past the point of where the reactant was. Now I've got my products. However, if I throw my enzyme in, then this energy is almost cut in half right here. So the energy required to start the reaction with an enzyme makes it much quicker to get to the same product. All right, so enzymes are reusable, as I stated with the enzyme substrate complex. Enzymes are reusable molecules found in living things. They end in ACE, again, ASE. So lactose is a carbohydrate that's broken down by lactase. Galactose is broken down by galactase. Fructose, fructase. Protein, protease. Lipid, lipase. So these are things that break down the carbohydrates, proteins, and fats or lipids. All right, enzyme vocabulary. Substrate, as I've been calling it, the substrate, right here, this yellow substrate. That is the molecule that enzymes work on. Products. Okay, the products are what the enzyme helps produce from the reaction. So when this comes out and it's broken into two new substances, that is the product. Active site, which is a very specific shape on the substrate and, on the enzyme. So it's the part of the enzyme that the substrate molecule fits into. It's like a puzzle piece or a key in a lock. Your house key isn't going to work on your car because it's got a specific shape that fits in there. It's shape that matters. If you look here, it says substrate binds to the enzyme. So this is our enzyme substrate complex. So here's sucrase, and my substrate is sucrose. It's going to come in and it's going to bind to that enzyme. Water molecules are added in hydrolysis, and the substrate is converted into products, fructose and glucose. Here, the products are released and broken down by dehydration synthesis. And then you have the active site is available on the enzyme for another molecule substrate to go in, and it's a cycle. That enzyme, unless it is denatured by temperature, pH, salinity, or something, or the enzyme concentration or substrate concentration changes, it continues to work at that same rate. Alright, so the lock and key model, as I mentioned, the shape of the protein allows enzyme and substrate to fit. Specific enzyme for each specific reaction. The shape matters most. So if that shape is damaged or the enzyme is denatured and changed, it cannot work any longer and do its job. Active sites. The enzyme molecules contain a special pocket or cleft called the active sites. I told you it's like a puzzle piece. Here's your substrate. There's your enzyme, active site, and there's your products after the substrate has gone into the enzyme and the enzyme has gone to work. Alright, so denaturing enzymes. When an enzyme is denatured, it means it's damaged. Denaturing changes the shape, and therefore if the shape changes, because of course we've been saying shape matters with enzymes. If the shape changes, without the correct shape, enzymes won't function properly. They won't be able to attach to their substrate. How are enzymes denatured? Two main ways, pH and temperature. Alright, what other factors control actions of enzymes? Temperature, water content, pH, chemicals, alteration of substrates, and alteration of products. Alright, enzyme inhibitors. I mentioned earlier inhibitors are things that prevent or stop something from occurring or decrease the chance of it occurring. Chemicals that prevent the enzyme from working. Inhibitors decrease the enzyme reaction rate. So certain drugs, pathogens, pesticides, and metabolic imbalances are inhibitors. Found in nuts, grains, legumes, and seeds. So right here at A, you have a reaction. The substrate comes in, attaches to the active site, goes through the process of enzyme binding, and then comes out in its products. However, if this inhibitor attaches to the active site, the substrate has nowhere to attach. So when it comes in, it's another form of competition. They're competing for the same resource. In this case, the resource is its enzyme. So the substrate and the inhibitor are the same shape. Alright, enzymes play essential roles in regulating chemical pathways, hormones, keeping us balanced, making materials that cells need, such as proteins and things like that, releasing energy, and transferring information. Okay, finally, the last biomolecule of the 4, nucleic acids. The 2 nucleic acids that we have are DNA and RNA, deoxyribonucleic acid and ribonucleic acid. They actually have the name of the biomolecule in it. So nucleic acids are macromolecules that contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay, in these specific elements, these 5 specific elements, are found in all living matter. Now, carbohydrates, lipids, they have CHO, carbon, hydrogen, and oxygen. Proteins have CHON, carbon, hydrogen, oxygen, and nitrogen. Nucleic acids have CHOMP, has them all. So all biomolecules have CHO in them. All living matter has CHOMP in them. Alright, introduction of nucleic acids. They're found in everything living. Number 1, everything in our world that is living has nucleic acids. It has the same universal genetic code, the same DNA. And I know that sounds weird. Yes, our DNA is different and nobody has the same exact DNA unless you're an identical twin, but that is not based on DNA itself. It's based on the sequence in which that DNA is written. Okay, so all living things have the same universal genetic code. The sequence in which that genetic code is written is the difference in species and organisms, plants, animals, trees, grass, fruit. All these things that are living, you're eating, they had DNA. All right. Monomer, again, is one part, is a nucleotide. It consists, so this is an example of a nucleotide right here. Okay. It consists of a 5-carbon sugar, or it's in the shape of a pentagon, so a 5-sided sugar, a phosphate, and one of 4 nitrogenous bases. So that's a nucleotide. One of those, this whole thing, is the one monomer. Okay, the polymer is DNA and RNA. They're made up of repeating nucleotides. DNA is deoxyribonucleic acid. The sugar is deoxyribose. And RNA has ribonucleic acid because its sugar is ribose. So it's the difference in the sugar in the name. Alright, repeated nucleotide monomers each contain a sugar, a phosphate, and a nitrogenous base. Again, so in DNA you have adenine, thymine, guanine, and cytosine. A bonds to T, apple to tree, and C bonds to G, car to garage. Okay, if you notice, there's 2 apples per tree. They have 2 hydrogen bonds between them. And there's 3 cars in one garage. They have 3 bonds between them. That's not necessarily super important right now as long as you know that there is a nucleotide and it contains a 5-carbon sugar, a phosphate, and a nitrogenous base, one of the 4. So each one of these little black squares is one nucleotide. So that would be the monomer. All of these together is your polymer or DNA strand. Right here. Alright, nucleic acids. Individual nucleotides can be joined by covalent bonds to form a nucleic acid. The function of nucleic acids, there's 2 functions. They store and transmit genetic information, DNA and RNA, and use to make proteins, RNA. There's not much else that we'll get into right now on nucleic acids. We will go more in depth in what the function of nucleic acids are in our next unit in cells, function and structure, and then even deeper when we get into protein synthesis later on this semester. So now in your journal, or on a loose piece of paper, whichever you want to do. You're going to draw a diagram similar to this. You'll have 3 different circles. Okay, the top one, put carbs, lipids, proteins, and then in the center right here will be what all 3 have in common. Okay, right here's what the green and the orange have in common, so your lipids and your proteins. Right here's what your proteins and your carbs have in common. Right here would be what your protein and your lipids have in common. Then out here it's what are the characteristics of just carbohydrates? What are the characteristics of just proteins? What are the characteristics of just lipids? Go ahead and do that and turn it in and move on to your next assignment. If you have any questions, please ask, and we'll see you in class.